that's why we call it the Bangalore. My husband and I are both deeply into Bangalore. This is the house my father built 50 years ago. Of course, it's changed shades, but they were living here on this for very long. And now we decided to open it to Bangalore. Not only do we host our own events, it's open to Bangaloreans to host their own events as well. And rent it out for free. So that's just what I want to let you know. And all of you who have registered for this event, you are now tracked, and you're going to continue to get information about.
What does it say? I can't read.
If you do not want to be,
Can you see?
Want to be spam, don't save our number.
And it's showing you.
It's
showing you. We need to show me.
Yeah, so let's turn the camera.
Yeah.
And unsubscribe if you don't know
And the
that.
direction I will. Yeah.
So once I stand there, you can.
Yeah.
The slides have to be seen.
Yeah, it's there.
Very interesting.
Is it on?
Yes, live.
So keep them regular, so even in the space, that's fine. So, now I want to say how delighted I am that I've got Narendra Chirule speaking here. For us, Narendra is like a part of our family and a very enthusiastic part of the family. So whenever he visits from the US, it's like fun and games, board games, let's do this, let's do that, let's go have coffee, let's see a film, late night film, all these impulsive things happen. So we often forget just how distinguished he is, actually. So and that's why it didn't even occur to me to invite him, because he's not here half the time. He's in Pune, he's 6 months in the US teaching at UPenn, 6 months here running his company, and that which is in Pune. And only once in a while he comes to Bangalore, and it's all fun times. I never thought of inviting him as a speaker until he said, look, this is my last Sunday in Bangalore, next Sunday I'm off, do you think? So I'm like, oh yeah, of course. And then poor man wanted to speak about genetics and everything, because he's into drug development, and he's written this wonderful book, really nice book called Good Genes Gone Bad. Huh?
Druggy.
He's total druggy and has been for 3 decades, yes. Yeah. So and so he's written this very nice book on drug development, how the trials happen and how many go so badly wrong, and how people have to just pick themselves up and go on. And so we take for granted COVID vaccine came so quickly, but the amount of heartburn and heartache that scientists go through, that researchers go through before that is huge. So that book gives a very nice account of all that. Apart from that, let me read his official bio instead of blabbering on. Narendra, founder of Symphony Technologies, a data analytics company. He currently teaches drug development at UPenn and also at IIT Bombay and Delhi. As former head of R&D at Biotechnology Bangalore and in leadership positions at Amgen and Merck vaccines, he has contributed to the clinical development of vaccines, biologics, and cell and gene therapies. During his academic career, he has worked on the development of a leprosy vaccine, the pathogenesis of AIDS, and gene therapy for several rare diseases. Dr. Chirule is on the NIH Advisory Committee for HIV vaccines and on BIRAC Grant Scientific Review Committee. Sorry, I don't know what that is, but it sounds very interesting, which I'm sure it is. He is a TEDx speaker and has published a book, Good Genes Gone Bad, by Penguin, which is brought out by Penguin Landmarks, both of which describe lessons learned from colossal failures in drug development. Now, he wanted to speak about that, I'm sure, when he suggested, and I said, well, we are the Bangalore room. So whatever you speak has to be connected to Bangalore somehow. So he had to change his whole talk. And so, because of that, we have a wonderful new talk by Dr. Chirule. Please welcome.
It's always fun to come here. Can you hear me at the back? Now.
You have to plug.
Yeah. We're live streaming this on YouTube. And I guess you can check with people who are looking to see whether it's correct. Alright, so when clicker is So when Rupa told me to talk about Bangalore in this room, I don't know whether I'm a Bangaloreite or not. I've been here for 10 years.
You're not, otherwise you said Bangalorean.
So it proves that I'm not. But I live, like you said, I live for 6 months in the US and I live for 6 months over here. And this can be a completely separate talk. I think this whole concept of nations and cities and everything is just farcical. It's only for the political system to become political, have political leaders and gain power and all that. Everything should be fluid. Everybody should be allowed to go anywhere they want and live a great life. And just that's what it should be about. And that's what I do, right? I live anywhere, wherever I am doing. So anyway, that's maybe for another talk and another thing. But what I thought I'd talk about today for about 20 minutes or so, maybe 30 minutes, is give you a brief overview of my view of what's happening in the field of drug discovery, biotechnology in general, and what a fascinating time it is to be alive. I think there's one slide where I want to get your feedback and I'll tell you about that. So it's the interface between biotechnology and information technology. What better place to talk about this than Bangalore? I took a picture from the internet. I think this is Brigade Road, or maybe 1960s or something, for those of you who probably were here at that time. And this is now, and this is what ChatGPT says a utopic Brigade Road would look like. No cars. Wouldn't that be wonderful?
Yeah.
So, I have a few things about Bangalore. So, Rupa's book about Bangalore, I've read a few chapters here and there when she published this work. And Rupa is such a fantastic author. And I think I've learned so much from her writing, the casual writing, especially from the children's perspective. And she's a very good children's writer. And I shamelessly use her methodology to write deep scientific talking. Hey, do you know about this gene editing technology? And I'm writing this in big manuscripts and all that. So what I'll tell you today about is the ITPT world, a bit of history, and my history of Biocon is primarily because I was at Biocon, and so a lot of the history I know is from Biocon's eyes, so to speak. I'll tell you about the revolution that's happening right now, imagining the future of what that could be, and one slide at the end about what's happening in the startup culture in biotechnology in Bangalore, which is just absolutely amazing. So let me start with the history of Bangalore and Biocon, biotechnology. It's all intertwined. And on the left side of every slide where it's relevant, I've given a reference to a book that you can read. Because it's Bangalore Room and Rupa is doing this, so I've learned that there's so much information that you can get about things from books, especially scientific things, that in the field of science, we are so busy reading papers, manuscripts, and we say, okay, we are reading so much, why should we spend time reading books? Books are prehistoric. By the time it's published in a book, the science is already outdated. Why read a book? But authors like Siddharth Mukherjee and Abdul Gawande, people have now started writing these very interesting scientific books which tell you the history of how the science was discovered. And it's only in the last 5 years that I've been starting to read these books where you learn the context of things and then the science becomes more interesting. So historically, Biocon started in 1978. Founders Day is 29th November. Kiran started this with the metaphoric starting in the garage at 10,000 rupees. Initially they were making enzymes for almost 20 years. Then in dramatic style, Biocon, Kiran has done this every 10, 20 years. She's revolutionized the business, sold a very successful enzyme making business and decided I'm not going to make enzymes anymore, I'm going to start making drugs or therapies. And they started working on statins, and today they're the world's largest statin manufacturers. Right? You don't imagine that this is such a big company making such a vast amount of statins that is supplying the entire world. Then 10 years, all of this it didn't finish, this still continues. Then in 2010, between 10 and 12, I joined in 2014. So around that 2010, one of the things that they wanted to move into was biologic drugs. And I'm going to talk most about, my talk is going to be about biologic drugs. A biologic drug is one that is made by a living system. So the simplest one would be something like growth hormone, which is a protein that all our body makes. the growth hormone gene, the human growth hormone gene, is inserted into a bacteria called E. coli. The bacteria has grown in a culture, and when that protein is secreted, because of the replication of the bacteria, growth hormone is purified, and that growth hormone is then put into a vial and injected into tissues. So that's a biologic drug versus a chemical drug which is chemically synthesized. Right? That's the difference between biologic and small molecule drugs. Insulin was the first one that Biocon made, and that in 2004 they got it approved in India, and it really revolutionized pricing. Even today, if you want to buy a vial of insulin in the United States, it costs $178 for a vial, right? And here I think it's like 100 rupees or something. Now it's 300 rupees. 300. But whereas, it's a factors of orders of magnitude very different, right? So, affordable medicines is what I'm going to talk about what's happening in India in a few slides later. But let me tell you about the Biocon story. Then in 2020, the biosimilars were being developed, and they continue to be developed. And what are biosimilars? Biologic drugs which were developed 30 years ago in the US, like I was telling you about growth hormone and monoclonal antibodies, which are transforming medicine. These are very curative medicines, were going out of patent. And now, Biocon and companies like Biocon led the way, are developing these biosimilars, which are now taking the market by storm. Again, making all of these amazing drugs very affordable for many, many countries around the world. And I'm going to tell you the final story, which is 2025 and beyond, which is the whole talk, most of Europe, is what's going to happen in the future. So that's sort of a very brief overview of what the Biocon story, very detailed in this book called Myth Breakers. Some pictures, it's amazing that from the 1990s, even earlier, you can basically all the Nobel laureates and everybody came to Bangalore and came to Biocon, gave talks. And I don't know whether it was publicized well enough or not, but James Watson, who's the discoverer of DNA, was in Bangalore. And if Rupa would have known, I think we would have had his talk here, right? But and of course Shah Rukh Khan. So he was helping us launch one of the drugs that was just approved. And of course, Professor Kalam and everybody who came here, there are thousands of pictures you can look at on the website. Biocon's contribution to Bangalore has been huge. And most recently, they've supported building the metro that Infosys and Biocon have funded that metro on that. I don't know which color it is, but whatever color it is, that's the one that is supported by them. But over the years, yellow. But over the years, the Rajendra Shaw family have donated a lot of things around Bangalore and made what we take for granted. Is there's a role that Rajendra Shaw herself, but also through Biocon support, made things happen in Bangalore. So huge contribution, which is unrecognized because she does it so casually. You go anywhere, there's some aspect of what she's done. She's given some money, like this Narayan Hridayalaya Hospital in Electronic City, and that was founded by her big cancer hospital, and it's been around for 20 years. We don't realize all of that. This is in 2017, the first approval of a biologic drug, which is called trastuzumab, for treatment of breast cancer. It was approved in Biocon by US FDA. This was the first biologic drug approved by the US FDA made in India. And I was head of R&D. Of course, I didn't follow the work, but obviously I had a huge role to play in this. That is Paul Thomas and Mrs. Kiran, and they're celebrating the launch of this thing. Getting an approval from the US FDA for a company like Biocon in India is a Herculean task, and even I think it's bigger than the Herculean. I don't know what a task bigger than the Herculean task would be, but it is that. It is a massive effort, and maybe that's the details of that are written in that book and my TED talk, and there are more things that I can tell you about that. I told you about what next for Biocon, and in addition to all the things that I've told you, which will continue, new companies, Bicara Therapeutics, which is just went IPO in the US and is $1 billion valuation already within 5 years. And Iminil, Kiri is here from Iminil. They are developing CAR T cells, and Reva is also, she used to be in that company, developing CAR T cells, which are again revolutionizing therapy. I'll talk about that in a little bit. Lots and lots of biotech companies in Bangalore. I think Arun is here somewhere, and Arun has been doing some research on biotechnology in Bangalore. And I'm sure you know the excitement of this area. And I'll tell you where the gaps are and where the opportunity is here. So that's my story of Bangalore. Okay, now I move on to the biotechnology revolution and what's happening now. And so before I go into the biotechnology, let me tell you a very brief snapshot of my understanding of what happened, what the pharmaceutical industry in India has been for the last 75 years, maybe since 1901. 1901 is supposedly the first Indian company, Bengal Chemicals, which was established in Kolkata to make chemicals. That was the first Indian company. And just from a historical perspective, 1901, that's when Vivekananda died, right? That's the year he died. And the Bengal School of Art movement started in 1901. And the small biotech industry started. Or not biotech, this chemical industry started. But relevant 1947 to 1980, those 40 years or so, or 30 years or so, there was a slow growth of biotech, of technology, of chemical industries for drugs. Most of the drugs were probably imported at that time. There were some companies who were making it. My sisters here, Anjali, we were in Kolkata and Lucknow growing up, and we used to hear about these. You go to the dispensary, and the doctor gives you this solution. What is it called? Compounder gives you this mixture. And you drink that mixture for anything, whether from a common cold to Nilima is here, and you and Viveka here, doctors who are with us in Lucknow. So that's what was happening in 1947 to 1980 or so. From 1980 to 2000, approximately, I'm just rounding up these numbers of these years, but just to give you a snapshot of the history. What happened was, driven by Yusuf Hamid, the founder of Cipla, and many others, like Prasad and others from Dr. Reddy's, there was a revolution in creating making very, very large amounts of these chemical drugs. The education system, from the biology chemistry perspective, was a phenomenal time for engineers, chemical engineers. IITs were coming up at that time. ICT Mumbai, at that time it was called UBCT. They were having these fantastic chemical engineers, and chemical engineering was a field which was growing exponentially at this time, right? In 1980 to 2000, even a little bit before that. And because of that, the ability to transform manufacturing of chemical drugs was fantastic. So I'll give you an example. There was a drug, statin drug that was developed by Merck at that time, which just let's say hypothetically, it had 25 chemical steps to get the final compound, right? Cipla made it in 4 or 10. Instead of 25 steps, they could make the final product with the same kind of quality in 10 steps. And they were able to do that very efficiently, and at the scale, because of the effort that they required, they could make it at a scale which was ginormous. Right? So that was the innovation that was happening. We might say, oh, they're just copying the drugs. Yes, they were not copying, but looking at the final product and making that final product here. But there was enormous amount of innovation that was going on to make that drug in such big volumes, which was providing to the rest of the world, right? 100 countries were being supplied at that time. Something happened in that time frame. HIV, the disease was happening at that time, and the drugs that were being made at that time, fantastic drugs were being made at that time. And these drugs, these were pills that were being given to patients, and these pills were very, very effective. They were effective in blocking HIV replication. But they were $100 a piece or more, right? Cipla made it in less than $1. But they infringed on the patent. And in the World Trade Organization, they won the patent. And that's why they were able to give this $1 drug to all of Africa and all these Southeast Asian countries. So Cipla has contributed to controlling HIV in that period single-handedly. You know, and then of course because of that, other companies also got involved in that. But then, you know, when good things are happening, people don't like it, or, you know, so what was happening from a patent perspective? In discussions with, at that time, the political system, Indira Gandhi was involved at that time. She had enabled the patent law at that time where you could have a patent, if there was a patent outside the US or outside India, there was the product patent was not recognized. If you can make the product using a different process, then you could circumvent that and make it. So if you have, it didn't matter if you had a product patent, as long as the process that you made it was different. And because of that, in these 20 years, all the companies that we know of today, Glenmark, Sun Pharma, which was at that time Ranbaxy's, Dr. Reddy's, Biotechnology, became very, very good at making very large amounts of these drugs through a process which was different from the product. And they were making all of these drugs. They didn't have to discover the drug. The drug was discovered by somebody else, right? But in 1993 and 2004, the World Trade we had to sign the World Trade Organization treaty, the Indian Patent Law Office, and then since that time, now we have to the product patent to recognize. But what had happened 20 years before that, it allowed the pharma industry to become this massive industry during that time. And now they can play on the same field as everybody else. And that allowed the Indian pharmaceutical companies to become so big. We always wonder, like, how is the Indian pharmaceutical industry called the pharmacy of the world? It's because of that. That's what the history of pharma industry. Then I told you about the biosimilars, which are basically the same thing as this generic molecule, but making biologic drugs are much more difficult. So that's what happens till 2020. Today, more than 40% of the drugs that are made in the United States are made in India. 40% of the drugs. So it's a huge percentage, right? Given so many countries are making these drugs. Going forward, it's going to be advanced therapies and novel therapies, and I'll talk about that in the next few slides. That's the revolution that's happening. So in the book that I've written, I talk about, from this perspective, I'll talk about how the industry was evolving. The analogy is like when we go to school. In 1947, we were probably kindergarten, less than kindergarten. Then from generic drugs, we probably went to high school, or middle school and high school. Then when we started developing our own generic drugs, then we went to college. Then we started going to college, right? And from a biosimilar perspective, we are now, let's say, the PhD level, from copying the drugs, let's call it, right? But we have never, I'm going to stick my neck out and say, we have never really developed novel drugs. We don't develop novel drugs. We know how to copy somebody else's. I'm just being very generic, but we know how to copy somebody else's drug really well. But if you tell me that we develop novel drugs, we don't do that. And okay, when I do this presentation, I say we don't develop novel drugs. Somebody in the audience will hey, but what about that Glenmark one drug that we got? Maybe there are 10 drugs, 20 drugs, right? There are 300,000 drugs in development in the US, novel drugs, right? At various stages. Then where is the scope? And do we have to really transform the way we have to develop novel drugs, and the opportunity in India is now, and it is because of this IT/BT sector. That's where the transformation in biotechnology is going to happen in the next decade. And that's the story I'll tell you. So what are the factors, what are the pros of why outstanding talent. Yes. Can I just?
What's a novel drug?
What's a novel drug? A drug that reduces cholesterol. I'll tell you more detail about it. It's a drug that blocks a protein called PCSK9. If you take a statin and if you have a cholesterol level of, let's say, 500, 120 is normal, right? It'll take you 6 months of taking this drug, and then it'll come down to maybe from 500, it may come down to 300 after 6 months. You take this novel drug with the novel mechanism of action, you have a cholesterol of 5,000, not 500, 5,000. Next day it comes down to 120. Right? These are kind of novel drugs with very novel biological mechanisms of action. We understand the biology. Thanks for the question. So, outstanding talent. I don't need to elaborate on this. You know why we have these fantastic educational systems. Plethora of international level standard institutions, right here in Bangalore, right, from Indian Institute of Science to NCBS to INSTEP. Growing economy, access to cutting edge technology, and government funding. BIOTech, by the way, is the Department of Biotechnology funding agency which funds entrepreneurs. What are the challenges? The risk appetite. We don't have an appetite for risk. Right? I'd rather open a chai shop than do something risky. I know if I open a chai shop outside my house, I'm guaranteed to make money. I might even make a huge profit out of it. But if I have to do something a little bit more riskier than that, I don't feel like taking risk. In biotechnology, the risk is very high. I'll tell you a little bit about the investor confidence and why there is less investor confidence in that. Very limited collaboration. In the academic, in biotechnology, I don't know how it is in the engineering field, but in biotechnology, there's collaboration sucks. I think, and again, in the audience, hey, but you know, Indian Institute of Science developed this drug with somebody else, and of course. But I'm just going to say that because now I can. For 5 years, I tried to have an MOU with Biocon, India's one of the largest biotechnology companies, with Indian Institute of Science. Unsuccessful. You cannot make an MOU with Indian Institute of Science. You can't agree on the IP clause.
What is the problem?
IP clause in intellectual property. So, Biocon's lawyers will say, if anything discovered in this collaboration belongs to Biocon, and Indian Institute of Science, no, it belongs to Institute of Science. We discuss what percent, 20%, 50%, 30%, 20%, and then I retire. So, anyway, so I'm just exaggerating a little bit, but I'm telling you from my lens that I think we could do much better in collaboration. And I'll tell you an example of good collaboration in a little bit. Innovative thinking culture during education, teaching curiosity to children. It is happening now. Maybe those of you who are in the education field may realize it a little bit more. But I think there's very little, even today I would say, and I do a lot of this teaching in many different institutions around India. I teach it, I also give these lectures in high schools and colleges, and so in the last 6 months I've given 45 lectures, right? So, because I love it, and I love talking to people. And so I'm telling you this concept of curiosity is not rampant. Right? There may be a student or two, 10 students who are curious about all kinds of things, but it should be taught. To be able to do research, you need to be curious. Right? So anyway, that's sort of the challenges. So what is the solution to all of this? And I think it's bridging this chasm between biology and math. And I think we all know that these two are, what the analogy, two rails of a train which never come together, la 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 la, right? But essentially, this is the space which I think India has an incredible opportunity to be able to transform the world. Right? And I'll give you some examples of this. So these are books, two books that have recently come out, one from Eric Topol and the other from Peter Lee, who's the head of science in Microsoft. Revolutions in AI and Deep Medicine, with excellent books. But when I talk about the space between IT and BT, it is this bioinformatics, use of machine learning, cloud computing, digital telemedicine. That's what I mean when I talk about the space between IT and media. So here's the kind that I'd love to hear from you a little bit. You can shout it out. Can you name some transformative innovations? It doesn't have to be in biology, it can be anything. What do we call transformative innovations that have happened in the last few years? Teflon several years ago, yeah?
GPS.
GPS, yes?
UPI.
UPI, very good. ChatGPT. Maybe in the biological fields?
The entire protein business with
Protein, yes, yeah. Looking at protein structure, very good. Here are some, there are many, but here are some that I pay attention to because of my interest. This is the AlphaFold. AlphaFold is the software that solves protein structure. Just to tell you the magnitude and how important this discovery or invention, whatever you call it, is. Before AlphaFold was available, this is in the public domain, by the way. In order to understand the structure of a protein, you have to make the protein first. Somehow you have to make this protein. You want to understand the structure of an insulin protein, you have to make it. You clone the gene, you have to make the protein, you have to make Gauss amount, milligram, kilogram amount of the protein. You make this protein. Then you have to crystallize it. And crystallization of protein is a very, very tricky process. It's a science and art. So you take this protein and you put it in various buffers, the right condition, the right temperature, the right altitude, and the right attitude, and all the right and then suddenly the thing crystallizes. And when it crystallizes, you write your paper to nature, I crystallize. And then through X-ray diffraction and then getting into different rays and the diffraction process, you get the beautiful structure of this protein, right? It used to take 10 years minimum, sometimes 50 years, to solve the protein's crystal structure. In a few seconds, it solved of 200 million proteins, and it solves the process in seconds. This is the power of the AI and the technology. This is the transformation of the technology. So now, if we can solve the problem, if we can solve crystal structures or get protein structures in seconds versus 10 years, what are you going to do with this data? We don't know what to do. We know. I'm not saying we don't know. This is what the kids we can tell the kids to do a tough job for us. We can just state the problem. Now that we have all this information, what are you going to do with it? How are you going to discover new drugs? How are you going to think about this? Whole genome sequencing. The first human genome was sequenced in 2000. Year 2000, it cost $1 billion. $1 billion in 2000 to do it. Today, it costs less than $1,000 to do it. In science, in computing language, we hear about this thing called Moore's law, right? But it's something like every 2 years, something speed increases, right? This has completely broken Moore's law. The ability to sequence DNA is just phenomenal. You can go and get, it won't be long before, I mean the next 5 years could be, as soon as that. You go to get milk, you can get your DNA sequenced. It'll be like that. It'll become like that. But now, just like the AlphaFold, I have my DNA sequence, 3 billion nucleotides. I know which ones 3 billion nucleotides I have. I make 600,000 proteins in my body. I know that. So what? So what? I don't know what to do with this data. Right? I need like I tell my friend in my company, we get this data from the research collaborations and all, huge Excel spreadsheet, 5,000 rows and 200 columns, and here's the data. Now look at it, right? And as a biologist, all I can do is I can squint my eyes a little bit and maybe is there a pattern? I can't see any pattern. As a biologist, I have not been trained to look at data. I've not been trained to analyze data at all. I get terrified with these Excel spreadsheets. Right now, a little bit less, but it's very good. And for those of you, all of you are engineers, most of you are engineers, this is sort of easy for you. But that's whole genome sequencing. IGIB, the Indian Genomic Institute in Delhi, which I visited, has now sequenced the first 10,000 Indian people's human genome. Right? This is 10,000 genomes are done. everyone knows about mRNA technology, gene editing, bioprinting organs, and de-evolution. You all have heard this, the fox, the two new fox, which were extinct. Woolly mammoth is going to come back. This is being done in MIT. So fantastic stuff in biotechnology is happening, right? Again, a little longer list, which is more personal to me. Something called a virtual cell. I'll talk a little bit about that. Organoids, organoidal models, growing organs outside the body, predictive data analysis using biotechnology to do drug discovery, and then mapping the human genome. So this is a paper that was published in Cell, How to Build a Virtual Cell for Artificial Intelligence. So it's a single cell, it is completely digital. Just to give you an idea of what we are talking about, there are 10 to the 10th proteins in a cell, 10 to the 9th carbohydrates, 10 to the 10th lipids, RNA, DNA, 10,000 genes are expressed, 1,000 transcription factors are expressed in a single cell. There can be mutations, the proteome complexity, there's a metabolome that is happening inside a single cell. In a single cell is huge, huge, right? This is digitized. With virtual cells digitized now. Every component is digitized. You can actually ask the cell to do what to do, not in a lab, but on the computer. This is the power of this technology. It's transforming, and this is happening at a pace that is ridiculously fast. Every week, every month in these journals called Nature and Science, there is a paper that will probably win the Nobel Prize every week. Such revolutionary work is being done. The pace is you can't keep up with the pace. Right? Let me just finish with the recording, and then we'll stop the recording and we'll do questions. And the last point I was saying, this came out just yesterday in Nature. In 1 millimeter square of the brain, they have made a 3D map, a digitized 3D map, right? There are literally trillions of interactions in that they've digitally mapped. It's only 1 square millimeter, right? They have to still do the rest of the brain. But this is just an example of Santiago Ramon Cajal in 1920, looking at the sections of the brain, drew a picture of the brain, and because he drew this picture, he got the Nobel Prize. Not because he drew the picture, but that was 1920. 2025 digitally, we will know every aspect of the brain digitally. Again, so what? It's a question of you're going to get all of this, and what are you going to do with this? So how are we doing with time? 15 minutes, okay. So, to put it in context, I've been telling you these random, grandiose things, fantastic, this, that, and the other, and they're like, okay, great, we'll go home and say, we'll put on, and say, okay, what did he tell us? No idea, just him wearing a check shirt and his shoes matched his shirt. Anyway, let me tell you a story of cancer, which Siddharth Mukherjee has told it beautifully in his book. I didn't make that picture here in the raw format. Thanks for coming. So the story of cancer, of course, is 5,000 years old, but let's start with about 1900. From 1900 to about 2000, this period. The only treatment that was available for cancer at that time was cut, poison, and burn. You know what they are, right? You don't know. Cut is what?
Surgery.
Poison is? Burn is? Radiation. Cut, poison, and burn was what? If you have cancer, that's what you got. Right? Then in 2000, around 2000, the human genome was sequenced. So now you have started understanding molecular biology. Now you have started understanding proteins that are involved in the pathogenesis of cancer. You understood that the p53 gene, that the gene that is involved in the cell cycle, and if you target the p53 gene, you can kill the cancer cell. So this is called targeted therapies. Right? That happened for 10 years. Then 2010, a revolution happened. Right? So till 2010 or so, if you had cancer, if you're lucky, you survived. But 5-year survival rate for any kind of cancer till that time was very dismal. Right? You would not hear a lot of people surviving a lot after 10 years, let's say. Now, with immunotherapy, people are surviving 20, 30, they're completely cured of their cancer. And these are cancers that will kill these kids who are being treated right now with the amazing growth. That's immunotherapy. I'm extremely biased because I'm an immunologist, so I'm telling you from an immunology perspective. The story really started a long time ago. This is Robert Schreiber, that's him shown here, University of Washington. He gave a very nice theoretical explanation of how cancer deals with, how the immune system deals with cancer. It's called, they're called the 3 E's of immunology, cancer immunology. I won't spend much time, but the basic theory is we all in our lives, normal immunologically, are getting exposed to all kinds of pathogens, sunlight, toxins, and our cells are mutating all the time. But our immune system is very strong. It can recognize these mutations and kill, and this is happening all the time in our body. At some point, what happens is the cancer, in order to evade the immune system, expresses certain kind of protein of itself to block the immune system from killing it. Right? And so, Jim Allison and Tatsu Honjo in 2010 discovered the cancer cell do this. They discovered, that the cancer cells evade the immune system by doing this, and for that they were given the Nobel Prize. And since then, a drug, many drugs, but the main one is Keytruda, which is made by Merck. They made a drug that blocks the inhibitory signal of cancer, and this drug is a miracle drug. You take this drug, cancer is cured. It's cured. You get cancer, you take this drug, but it only works in 10% of the people. 90% of the people, it really doesn't work well. 90% of the people get relapsed. But 10% of the people completely cured. This drug is very expensive. $200,000 per year. Wow. Right? And this is where the biosimilars come in. So when Indian companies will start making biosimilars, it will become cheaper. Keytruda makes something like $30 billion a year or something like that. A very dear friend of mine works, is the head of marketing for Keytruda, so we have a lot of debate about pricing and all, bantering all those things.
It's about 8 lakhs for 3 doses.
8 lakhs for 3 doses. So then, fast forward, just to give you an example, these are Kaplan-Meier charts. On the y-axis is the survival probability. Just think about it. And the x-axis is time. And you can see that cancer patients, in general, if you don't treat them or treat them with the regular drugs, cut, burn, poison kind of drugs, they will die after a period of time. Right? But if you treat them with the immune checkpoint inhibitor, that are these drugs that I just showed you, their lifespan is expanded. So the phrase that is used in cancer immunotherapy is to flatten the curve. Flatten this curve. So if you flatten it, you're basically curing cancer. And the last one that is shown here in red is called CAR T cell therapy. There's time in the question and answer, I can explain that in detail, but we have folks from India who are working on this right now here in Bangalore, again, transforming how cancer is being treated. And then finally, bringing back to ITBT, there are so many factors, input parameters that are there for cancer. Only 10% of the people are responding. The question is, why are the 90% not responding? So the ITBT space, analyzing why are the 90% people not responding is a way to is an opportunity to study. And so, fast forward to 2020 for cancer, it has now become personalized therapy. Now you know the genome, you know the whole genome, you know which cancer is going to be affected, and you can make a specific drug for you. Okay? So now I'm going to digress a little bit. I told you about cancer, and I'm going to tell you about the imagining the future. This take a few minutes. And Dr. Pai at the back, we call him Anparna, he has inspired me so much to think about physics. I think it was almost 10 years ago you gave me that book from Edge of Life by Jim Khalili and John McAdams. Quantum biology, right? The space between quantum physics and biology. And I'm 10 years already, I'm still reading it and I still don't understand it. But I learned something from physics. So this is Ashutosh Gopal. Relima, Anjali and I, he's 5 years junior to us from school. So So he's one of our guys, right? So he's a particle physicist in Duke. I just have to tell him, because I'm so fascinated. I'm like in love with him or something right now. Because he's such a nice guy, right? Minimum, the cute guy. So he went to the best schools in India. Came first. He said he came first everywhere in school. Got full scholarship to do his undergrad in UPenn, and because his father was an electrical engineer, he also did electrical engineering. While doing electrical engineering, he also did a bachelor's, master's degree at the same time for boarding school in economics. Same time, right? What that? Why not? Right there, right? Then because he didn't get an internship in electrical engineering, one of the professors told him, "Why didn't you try the physics department? Because they seem to have funds." So he went and did physics, and he went into nuclear physics. And he started asking very basic questions to the professors. Right? And one of his professors' name is Fortune, Professor Fortune. So he said, "You're asking two fundamental questions. Nuclear physics is not for you, it's too general. You should go into particle physics." So he started studying particle physics, and then he got into Harvard to do his PhD. With like the god of particle physics at that time. Right? You can read it in his book. This book. Then he does a few postdocs here and there, goes and works, and now he's now turning 60 soon. Imagine that. He's a professor at Duke, and he just wrote a paper a couple of months ago where many of you have heard about the Higgs boson. He, through his methodologies, has defined the weight of the Higgs boson. This is an astronomical book. This is not astronomy in the English language. But if that was, it took 40 years, theoretical physics, it took 40 years to prove the theory into actual practice, right? And I was telling you about collaborations. That paper has 1,300 authors from all over the world, and they all work together. How can that be even possible? Right? At least two people should fight.
It's a God particle.
No, it's a God particle. That's why. Right? Okay, this fantastic story, right? This book is written by who, you know? His mother. She's a theatre artist. And she wrote her son's book on particle physics. Who are these people, right? Amazing. A very readable book if you've not read it.
What's it called?
Beyond the Higgs boson. We've got it at home if you want to borrow it. Anyway, the reason I'm telling you about this is, of course, we're going to talk about Ashutosh, but in biology, in physics, one of the things is there is a lot of theory. You know, you speculate and you think about this, and Isaac Newton does that, and so and so does that, and Erwin Schrödinger thinks about these things. Erwin Schrödinger in 1927 predicted what DNA would be, right? In his book, What is Life? In biology, at least in my reading, we don't have theory. We want to see data, and then from the data we will say, okay, this might happen. I think we need to move to theoretical biology. Oh, what am I? Why would I do this? Ashutosh just last week was given the breakthrough award. This is now the Nobel Prize of the US. It was given this prize last week. The breakthrough prize.
It is 13,000.
All 13,000.
Now 13,000. No, in the paper there are 1,300. In the collaboration of 30,000 people. And I don't know if all 13,000 will get it or not, but at least he's one of the 13,000, so I put his name. I checked with him the day he got it, and he did. So breakthrough prize is literally the Nobel Prize of. It used to be the last. It is still the last term, but breakthrough has become one of the most important prizes in the US, and he got that last week. We are also doing that kind of work in this room. Pranav told us about the quantum of riddle of life, and in this very stage where he was showing us how electrons are waves and particles, right? He was showing us when we did this, and so this concept of biology, physics, chemistry, and all that is coming together, and the Bangalore room is making it happen, right? And so a lot of things in theoretical biology that have happened. It's not like nothing has happened. But I think we are going to a digital youth. I think I told you a cell. Ultimately, we will have, all of us will be digitized at some stage. And so the final thing that in this slide about cancer, I'll tell you, and this is a beautiful set of papers that have come out with Cell, the journal called Cell. It is high impact factor journal. The first paragraph are really interesting. For the last 5,000 years, forget 5,000, let's say for the last 100 years, when Western medicine, it's called Western medicine, has been focusing on disease. So you have people get sick, and researchers try to find out why people are sick. What they are saying now, and you've probably been reading this kind of literature now, it's actually more interesting. Why are we healthy?
Yeah.
It is so interesting that we are healthy, and we don't study that at that level of detail like we study why we are sick. And I think what the focus in the future is going to be in medicine is the focus on health, not necessarily disease, so that you don't get sick in the first place. Right? And the last slide is a shout out to some really, really good, very, very innovative biotech companies that have started in Bangalore, which are doing some phenomenal work. And I can talk about each one of them in a whole lecture. We can maybe invite, and Rupa, maybe we can invite some of these folks to talk about their work here at some point.
Yes, we have made this like
Yeah,
this.
they are definitely Bangalore people. And so the opportunity is huge in biotechnology and drug discovery, diagnosis, precision medicine, monitoring. And final slide, Bangalore continues to be a leader in biotech and IT integration. Of course, I don't say that lightly, it's absolutely true. Innovation, government support, talent, driving processes, and future development is in biotechnology, it's in the space between biotech and IT. Thank you so much. Now we can do Q&A. If not the lights in the audience.
So how accurate is the virtual cell?
One minute, coming.